Dr. Christine Charvet is an assistant professor in the College of Veterinary Medicine at Auburn University, specialising in comparative neuroscience using big data in genetics and neuroimaging. She's renowned for her work on translating time, a resource that enables researchers to find corresponding ages across human and model organisms, and has actively led initiatives to promote diversity in biomedical science. And Dr. Christine is with us here. How are you today?
I'm great. Thank you for having me.
So, just before we get on with some real questions, what does all that actually mean?
Our work is focused on harnessing information about model systems, and we're using that to generate biomedical resources like translating. So basically, you can find out how old a mouse is in human days, and we're working on that and expanding that research.
So, does that mean like when somebody says dog years, it's kind of you're making it relative?
Yeah, it's relative, and there's still a lot of work to be done in that regard. What we're doing here is we're integrating different kinds of information, so gene expression, anatomy, behavior, and using a lot of information to figure out how old an animal is in human days.
Animals that live less than us, does it feel to them like a longer time, if you see what I mean?
Yeah, that's a fascinating question. We don't focus on that specifically, but we can definitely see that certain phases are accelerated in some species relative to others, but their perception or what is going on in their mind is a little bit more elusive to us.
So, how did you come up with the idea for translating time?
It was actually not my idea. It's an idea that started 25 years ago. So it's actually before my time. And it's a professor at Cornell University who basically in the 90s came up with this notion that you could use the timing of certain developmental processes to find out how old mouse is in human days. A paper published in Science. And so I worked with her at Cornell and I started my lab. And what I wanted to do with the Translating Time resource is integrate different lines of research into it, looking at multiple scales of biological organizations so that we could translate time across the entire lifespan.
Because you've mentioned a couple of times now how old a mouse is in human days. How come it's measured in days and not years, like, because that's the way you'd normally say how old someone is? Is it just because it has to be specific?
Yeah, so much of our resource is focused on prenatal development, so before birth even occurs. And so we use days after conception. Another reason is that when you look at neural events, you see that the timing of birth is all over the place, but the timing of neural events is more like conserved, or the pace and sequence between species is more conserved. So if you look, for instance, at some altricial species, so some species that are born in an immature state, like mice, and if you look at like a sheep or a goat, they're born in a mature state. They're like ready to go within a few days. So that's that age of birth is, it has a heterochrony. It's less predictable from the timing of other events. And so for us to be precise, we use days after conception.
So how is it that researchers actually use translating time?
Yeah, so that's a great question. So our resource primarily focuses on model systems. So we find corresponding ages across about 19 model systems in humans on our website called translatingtime.org. And when we mean model system, we mean an animal that is often used to understand human biology or in health and also in disease. And so the common model system is predominantly mouse. Other model systems are used as well. And so we have that range of model systems represented in our resource. And so it's mostly for biomedical scientists who are interested in solving human diseases. So they're interested in how can we use a mouse or a rat or a monkey to understand schizophrenia, autism, Alzheimer's disease. And we know that these diseases in humans occur at specific ages. So schizophrenia is more likely to occur during adolescence. Alzheimer's, is a devastating disease that impacts the brain and cognition, and that occurs at late stages of human life. And so in order for the mouse to be most useful to understand human biology, we need to find out how old that mouse should be for study. And so people will often use our resource to figure out, well, how old should that mouse model of schizophrenia be, or another disease that impacts developmental biology in humans.
Yeah, that's quite fascinating, because maybe a lot of people, when they first hear about this resource, would maybe think, why would you want to do that other than just for fun? But actually, it's really important, because I guess sometimes it works for things that aren't age specific, but you can't just test something on a mouse without necessarily knowing how old it is a lot of the time.
Yes, that's exactly it. So there are a lot of things, biological processes that occur at specific ages of our life. And so really use that mouse model to understand human disease. We need to find out what those comparable developmental processes are happening at those ages.
And how come it is always mice that's used? Because the most similar animals to humans are like apes and stuff like that. You wouldn't necessarily think of mice.
Yeah, so that's a great question. So mice are often used because they're easy to handle. They can be bred in large amounts. You can study large samples of them and study them in the lab. That's really not the case for, chimpanzees and orangutans.
Yeah.
And there would also be ethical issues, when studying some of our closest relatives. So mice were chosen because they were easy to handle and because we could genetically manipulate them.
From all the studies that you've done, what are some of the key findings that you've found?
Yeah, so we've worked on expanding this translating time resource. We've used different kinds of data like gene expression, anatomy, behavior, and using all that information, we were able to find out how old a human is in chimpanzee or gorilla or orangutan days. So we have expanded that resource to the lifespan. And one thing that's really striking from these, comparative analysis across the entire lifespan is humans have this phase at late stages of life, like 80-year-old, 90-year-old humans, with no clear counterpart in chimpanzees or other studied great apes. And that's because chimpanzees will live up to about 40 years of age. You'll find a couple of super agers, but for the most part, they don't live really past 40 years of age. And that maps onto humans in their 50s. And so, beyond that, there's no clear counterpart for chimpanzees. And what that means is that a lot of things that impact us at late stages of life, like Alzheimer's disease, which is, a disorder that impacts cognition and brain, is something that's rather elusive in those chimpanzees and other primates. So I would say that the thing that's really striking to us right now is the fact that late stages of life really, it just doesn't have a clear counterpart in those studied primates.
How come they don't get Alzheimer's and dementia?
Well, so our studies are finding that a 40-year-old chimpanzee is actually a 50-year-old human. We take a bit longer to age than chimpanzees, but if you look at the age translations, basically the 40-year-old chimpanzee mapping onto the 50-year-old humans, if they were to live longer, if chimpanzees lived to 70, that would maybe be 80, then we would start to see those aged phenomenon that we see in humans, but not so much in chimps.
So what are the factors that lead to a chimpanzee at 40 being a 50-year-old human? And how do you come to that conclusion?
Yeah, so we collected a lot of different time points. So we looked at gradual changes in biology and behavior, looking at transcription of the brain, looking at brain scans, and looking at these trajectories. So for instance, a gene expression will go up and down, and the correspondence in human also goes up and down. And we can use all of that information to find out that correspondence between a human and a chimp. And then that's how we come up with it. The fascinating question is, why is the clock different, between humans and chimps, and that's something that is a fascinating question.
Yes, it certainly is. Now, you are also looking into companion animals, especially cats, at the moment. So how do you hope this will enhance our understanding of human biology?
Yeah, so the thing that was really striking to us comparing humans and great apes is that fact that there's no phase of life in chimps. But when we look at companion animals, and in particular cats, we see a lot of those aged phenomenon, like cats have plaques and tangles, which is something you see in Alzheimer's disease. They live long lifespans relative to their wild counterparts. And some companion animals will have a disorder called cognitive dysfunction syndrome, which is basically something maybe akin to dementia in humans. So things that really impact humans at late stages of life are also seen in our companions. And in particular, we're studying these cats. And so we are looking to cats, thinking that they might be a good model system for human aging.
Yeah, well, they certainly must be, because I had a cat that had dementia once and it pooed all over the house. So I can testify that they certainly can get dementia. It's certainly interesting that all these different animals have different things going on. Is it like an evolution thing, do you think? Like maybe one day far in the future, if some things happen, then some animals will start to like get older, have an older human age or younger?
Yeah, so that's really interesting. The way we're thinking about it is these companions, these cats and these dogs, they're getting veterinary care. So maybe your cat got a lot of veterinary care, right? Because they're our companions. We love our companions, and we'll make sure they go in maybe for their wellness exams every year, take good care of them. And I think that good care is promoting long lifespans in at least some of the companions that we have. And so I think that this long lifespan in those companions is kind of a consequence of us caring so much for them. And I think in the wild, for instance, cats maybe are less likely to live as long as they do when they're in our care. So we see it as environmental effect, the fact that they live with us, share similar environments, and also are subject to medical care. And that's all promoting their long lifespans and leading to sort of these issues that we have during aging.
What are some of the ways that people can get involved with the research if they want to?
Yeah, so we have a project. It's called Project Catage. And you can find out more on my website, charvetlab.com, where we are actively recruiting people to donate information about their cat. So you can participate if you'd like, because it sounds like you had an old cat.
Yeah, I suppose. Does it count for dead cats as well as ones that are still with us?
So we want the veterinary record. So we want to know, we want cats with known ages, and we just want to go through their health records, and we're going to go through their health records so that we can predict how old those cats are in human dates, and we'll integrate that into the Translating Time website. We would be thrilled to get cats from the UK because we're really interested in getting diverse cats, because we want to see diverse cats and compare them to, the broad range of humans we see around.
Is there any other research projects you're working on at the moment or thinking about in the future, or is it all cats just now?
So cats were just something that we're really interested in because they showed those phenotypes, those aged phenotypes like plaques and tangles and extended lifespan. But we are actually studying a broad range of mammals, cats and then dogs, primates. And what we're really interested in is harnessing the diversity across life forms to inform human health.
Well, where can we keep up to date with the research and, of course, get involved, if you just remind us of all that?
Yeah, sure. So our website, our resource to translate time is called translatingtime.org. You can leave us some feedback on charvetlab.com. That's my lab's website. And so those are two resources where you can learn more about what we're doing and keep up to date and participate your cat in Project Catage.
Excellent. Well, many thanks for joining us. It's been excellent to talk to you.
Great. Yeah. Thank you so much.